Hello friends, today's topic is production of insulin hormone. Insulin is discovered by the scientists Dr. Frederick Banting and Charles Best in 1921. This hormone produced and secreted by the beta cells of pancreas, islet of Langerhans. Diabetic patients whose elevated sugar levels are due to impaired insulin production have been treated with insulin produced by recombinant DNA technology. Chemically, insulin is a small, simple protein consisting of 51 amino acids, 30 of which constitute one polypeptide chain and 21 of which comprises second chain. Both chains are linked by a disulfide bond. You can see the image 3D structure of insulin and chain A and chain B, which are linked by disulfide bond. For the production of insulin, weakened strain of E. coli it has been taken, which is a gram-negative bacteria, which is continuously being used in the genetic engineering of insulin. Steps of insulin synthesis. First step is isolation of pro-insulin gene. Gene responsible for the synthesis of insulin are extracted from pancreatic cells. In the first step, the genetic information for the synthesis of insulin transcribed to mRNA, meaning thereby that DNA is converted into messenger RNA. Then by using an enzyme reverse transcriptase, that mRNA it is supplied with the necessary nucleotides to produce a copy of complementary polynucleotide strand resembling the DNA strands coding for insulin. That single stranded DNA obtained is converted into a double stranded helix, and then double stranded DNA produced is called complementary DNA. This cDNA thus formed is called proinsulin gene. That proinsulin gene is linked to lac operon with B galactosidase, which provides the initiation and termination signals for the synthesis of insulin. Second step, that is insertion of proinsulin gene into plasmid. PBR322 of E. coli, that plasmid is used as a vector or a carrier. The restriction endonuclease enzyme called like HIND3 used to cleave the plasmid. And then proinsulin gene is mixed with cleaved plasmid in the presence of enzyme DNA ligase. The restriction enzyme breaks the tetracycline resistant gene of plasmid. The proinsulin gene is inserted into this region. Now the plasmid with inserted proinsulin, it is now it has gone for recombinant DNA. Third step is transfer of recombinant plasmid into host cell. E. coli bacteria are generally used as a host cell in insulin synthesis. They are treated with cold calcium chloride and the temperature is immediately increased to 42 degrees Celsius, resulting in destabilization of cell membrane and development of pores in that. Such cells are mixed with recombinant plasmid, resulting in the migration of recombinant plasmid into the host cell that results in bacterial transformation. This is the image of transfer of recombinant plasmid and of cloning. Four-step cloning of proinsulin gene. Before cloning of gene, the E. coli cells with recombinant DNA, it must be known, they can be easily identified because they are sensitive to the tetracycline antibiotic. Because In recombinant plasmid, tetracycline gene is damaged and contains proinsulin gene. Because that proinsulin gene is there, it has been located on that part or inserted on that part. Such E. coli cells are selected and grown in agar medium to get large number of similar cells with recombinant plasmid. Results in cloning of proinsulin gene, which are then used in insulin production. Fifth step, insulin production. E. coli cells with proinsulin gene in their plasmids are grown in bioreactors containing nutrient medium. Lactose has been added into the bioreactor that increases the production of proinsulin. It acts as a inducer, activates the lac operon linked to proinsulin gene. The product is treated with cyanogen bromide to remove beta galactosidase protein. Proinsulin naturally folds, so the cysteine residues are opposite each other, and the chemical treatment then causes the formation of disulfide crosslinks. Once this has been accomplished, the connecting mid-chain C can be removed by treatment with proteolytic enzyme trypsin and carboxypeptidase B, which have no effect on insulin itself, resulting in the formation of active insulin. These are the steps in insulin synthesis, as already mentioned or told. So advantages of recombinant DNA technology: large scale production can be carried out, high purity and quality of the product, rapid absorption of the product, less allergic reactions, and lower risk of infections. So that's all from my side. Thank you, friends. These are the references. Thanks a lot.